Now, let's look at how pH can affect the activity of an enzyme. To do this, we're going to set up a simple experiment using starch and different pH conditions. First, we label 5 test tubes from 1 to 5. Into each test tube, we add 5 cm³ of a 1% starch solution. This will be the substance the enzyme acts on in each case. Next, we adjust the pH in each tube by adding either an acid or an alkali, depending on what's needed. For example, in tube 1, we add 1 cm³ of sodium carbonate solution, which makes it quite alkaline, around pH 9. In tube 2, we use a smaller amount, 0.5 cm³ of the same sodium carbonate, making it slightly alkaline, around pH 7 to 8. Tube 3 is our neutral condition, we don't add anything to it, so its pH stays around 6 to 7. Then we move on to the acidic conditions. In tube 4, we add 2 cm³ of ethanoic acid, also known as acetic acid, which makes it slightly acidic, around pH 6. And finally, in tube 5, we add 4 cm³ of the same acid, making it strongly acidic with a pH of about 3. One important thing to remember, when switching between adding acid and alkali, we need to rinse the syringe thoroughly. This prevents mixing of chemicals that could change the pH and affect our results. By comparing how the enzyme works in each test tube, we'll be able to see how pH levels can influence enzyme activity, whether it speeds up, slows down, or stops the reaction altogether. Then, to continue the experiment, we prepare a cavity tile by placing drops of iodine solution into several of its wells. Using a clean syringe, we then add 5 cm³ of 5% amylase solution to the first test tube. After gently mixing the contents, we start the timer. Every 30 seconds, we take a small sample from the test tube and place it onto one of the iodine drops on the tile. Again, rinsing the pipette with water between each sample is essential to maintain accuracy. We continue sampling until the iodine no longer turns blue-black, indicating that the starch has been completely broken down into sugar. At that point, we record the time taken. These steps are repeated for each test tube, typically stopping after about 15 minutes. As the starch breaks down, the color of the iodine changes from blue-black to orange-brown. The faster this change occurs, the quicker the enzyme is acting, which reflects higher enzyme activity. We repeat this entire process for test tubes with different pH levels to observe how pH influences enzyme performance. To check the actual pH of each solution, we can place a drop onto pH paper or mix a sample with universal indicator in the cavity tile and compare the resulting color to a pH chart. Ultimately, this investigation helps us determine how pH affects enzyme activity and identify the pH level at which the enzyme, amylase, functions most effectively. This method provides a practical way to explore the relationship between pH and enzyme efficiency. As shown here, this setup is designed for an experiment investigating how pH affects the activity of an enzyme. There are 5 test tubes, labeled 1 to 5, with a syringe positioned above them. Each test tube contains a different chemical to create a range of pH conditions, as outlined in the accompanying table. In test tube 1, there is 1 cm³ of sodium carbonate solution, resulting in an alkaline pH of around 9. Test tube 2 contains 0.5 cm³ of the same solution, making it slightly alkaline with a pH between 7 and 8. Test tube 3 has no added chemicals and serves as a neutral control, with a pH of approximately 6 to 7. On the acidic side, test tube 4 contains 2 cm³ of ethanoic acid, giving it a mildly acidic pH of about 6. Finally, test tube 5 contains 4 cm³ of ethanoic acid, creating a strongly acidic condition with a pH around 3. In the next step, 5 cm³ of starch solution will be added to each test tube from a beaker, ensuring all tubes receive the same amount of substrate. Then, 1 cm³ of amylase enzyme will be added to each test tube, and the timing will begin immediately upon addition. This is crucial for measuring how quickly the enzyme breaks down starch under the different pH conditions. A spot plate or dimple tray, as shown, will be used for testing the samples with iodine to monitor the breakdown of starch. And, the drops from each test tube are tested with iodine solution, which turns blue-black in the presence of starch. This color change helps us determine whether starch is still present or if it has been broken down by the enzyme. It's important to rinse the pipette between each sample to prevent contamination and ensure accurate results. Overall, the diagram provides a clear visual summary of the varying pH levels in each test tube, the addition of starch and amylase, and the method used to track starch breakdown. So in this experiment, we were looking at how pH affects the activity of the enzyme amylase, which breaks down starch. We used 5 test tubes, each with a different pH level, from strongly acidic to strongly alkaline. After adding the same amount of starch and amylase to each tube, we used iodine to test how quickly the starch was broken down. What we observed was that test tube 3, which had a neutral pH of around 6 to 7, showed the fastest breakdown of starch. This tells us that amylase works best at a neutral pH, which makes sense because that's close to the natural conditions in the human body. Test tube 2, which was slightly alkaline, also showed good activity, but test tube 1, which was more alkaline, had much slower results. On the acidic side, test tube 4, which was slightly acidic, had moderate activity, while test tube 5, which was strongly acidic, showed almost no activity at all. That's because enzymes like amylase can get denatured, meaning they lose their shape, when the pH is too extreme, and then they can't function properly. So overall, this experiment clearly shows that pH has a big effect on enzyme activity, and maintaining the right pH, close to neutral, is really important for enzymes to do the job well.